Hey guys, my name is Tyler Swinehart, and this is my building biology project on gel electrophoresis. Gel electrophoresis is a technique used by scientists in order to separate charged molecules, for example DNA and RNA, to execute DNA fingerprinting. During gel electrophoresis, restriction enzymes code for certain base pair sequences. For example, they might code for the sequence GGCCA, and it will cut the DNA at that point. The result of these cuts are variations in the lengths of the DNA. We now have long, medium, and short lengths of DNA. This is the contraption where gel electrophoresis takes place. First off, it's important to note that DNA has a negative charge, which will result in DNA getting pulled towards a positive end. The way this contraption works is that the DNA gets placed into the small rectangular slips near the negative side. Then, the light blue liquid serves as a buffer and the electricity is conducted into it. The energy from the electricity pulls the DNA towards the positive end. It's also important to note that the smaller fragments of DNA move quicker than the larger pieces, which will result in a variation of the ladder-like bars of the finished product. So what exactly is DNA fingerprinting? Very simply put, DNA fingerprinting is a method to identify someone based off their DNA by looking at patterns such as DNA sequence or DNA length. DNA length is determined by gel electrophoresis. Gel electrophoresis makes DNA fingerprinting possible in the sense of DNA length. Since smaller fragments move quicker than larger fragments, they end up further down or further towards the positive side. As you can see, we have a mother, offspring, and 3 male contenders. Gel electrophoresis has been done on all of their DNA, and we now have each of their fingerprints done. For determining parenthood, half of the mother's DNA and half of the father's DNA must match the offspring's DNA. In this example, male 2 is our offspring's father. DNA fingerprinting can also be done to help solve crime scenes, as shown in the picture on the right. Gel electrophoresis has been done on the DNA from the crime scene and the DNA from all 3 suspects. In order to be proven guilty based off of DNA, they must match 100%. So in our crime scene, suspect 2 is the guilty party. Now on to my article. In my article, scientists are using DNA fingerprinting to determine the origin of pearls and gems. These precious gems are not found deep below the surface of the ground and can't be mined. These precious gems are made by living animals. Certain species are restricted from gem and pearl use, but poachers tend to not abide by these restrictions. For example, an elephant's tusks, ivory. Though they are precious, poachers are not allowed to kill them and take their tusks. The scientists from my article are trying to find a way to use DNA fingerprinting to determine where the ivory came from and help reduce poachers. Up until 2013, pearls and gems were known to not have DNA, but a scientist by the name of Mayer found DNA in these gems and pearls. At first, a 1mm drill head was used to make a small hole in the nacre and extract some sample material to be used for DNA gel electrophoresis. Since most pearls were drilled for usage, it committed no real damage to the overall pearl. The research by Mayer showed that in most cases it was possible to separate the origin pearl oyster species. Current research by the author shows that DNA fingerprinting can be adapted to other species as well, such as developments and techniques to determine the origin and species identification of elephant ivory using DNA. Though the intentions were good, this has so far been performed in no destructive way, because it requires a relatively large amount of genetic material. The goal is to find a less destructive method like that used by Mayer with pearls, which, hopefully, could be adapted to ivory, which would allow for smaller amounts needed to test the DNA. All in all, it has been proven that DNA fingerprinting is possible in pearls and gems, but ivory requires a large sample size, which does not help when only a jewelry amount of that substance is available. There's still research going on to try and reduce that sample size needed. Thanks for watching, guys.